is going to be strictly anatomy. Actually, oh no, I've got being recorded. Okay, there we go. So we thought this would be a useful exercise because mostly what we see on from the Allen Brain Atlas, and Mike Bienkowski will be talking about this a little more, gives the sagittal sections and the coronal sections from the Allen Brain Atlas. We took on this proximodistal and dorsoventral axes because, or rather the dorsoventral axes as something that hadn't been done. But also for those of you who are not currently working on CA2, I thought this would be particularly useful in how most of us are defining it. And let's see. Okay, there we go. So really, I think some of us in the field had noted that CA2 was molecularly distinct part of the hippocampus, and through the years, there had been a few papers on FGF and NT3, some neurotrophic factors that were enriched in an area that probably was CA2, but it was really the work of Fred Gage, Ed Lean, and Fred Gage's lab and the Allen Brain Institute that really got us thinking that the CA2 was a molecularly distinct part of the hippocampus. And that really started allowing us to really, well, of course, genetic access to do new and interesting fun things, but also just allowed us to explore a little more on this part of the brain. Again, some of the fun tools we've had now, this happens to be a GFP driven under the Amigo2 promoter, and then the mossy fibers showing here. What became evident with some of these tools as soon as people started double staining with the mossy fibers, like what Kohara had published, really kind of went at odds with the classical definition by Lorente Deneau. So if you see, just by definition, CA2 was the part of the hippocampus where, if you can see my cursor, where the mossy fibers end, and then before we get into the small neurons of CA1. And so that obviously didn't match up. And so really, was that CA2? We had noticed that, but I was a new investigator at the time. I wasn't willing to come out and say that this was CA2 with the mossy fibers, but Tonegawa was willing to come out and say that. But looking back, we see that Ramon Cajal actually had some drawings that resembled what we actually seem to observe. That is, the CA3 neurons have these thorny excrescences here. He drew two neurons labeled C, and I guess I was actually referring to the axon, but that either don't receive this mossy fiber input or do. And I think that these drawings more accurately depict what CA2 is. Now, I'm going to talk a little bit more about these thorny excrescences here in a little bit. But we also know that these molecularly defined CA2 neurons have a separate output, a different output that's distinct from CA3. And this was shown by Kohara also in Tonegawa's lab, was that they project to this deep CA1 layer. And these are the calbindin negative neurons for anybody following that. Also the inputs from the SUM, and I'll talk a little bit about that. We use VGLUT2 as a stain for those terminals. I think people from Rebecca's lab will be talking about that a little bit. Now, we got into this looking at LTP as some of these molecules were potential regulators of plasticity. No matter how hard we tried, we couldn't get LTP in CA2 compared to CA1. We weren't very sophisticated at the time with filling cells and then staining, but we did have some indication again that we were in CA2, and our reviewers, of course, were not kind when they said, how do you know you're in CA2? Well, we don't, except that we would get LTP if we weren't in CA2. So, maybe it's a circular argument, I don't know, but now we do have ways to fill the cells and double stain and say, yes, we're in CA2. We've done a lot of work on these regulators of synaptic plasticity in the pyramidal neurons, and I'm not going to have time to talk about them here, other than that some of these are the markers that we use. Zaf Bashir is also going to talk on some of our work with the mGluR receptor dependent LTD, which relies on some of these molecules. As an aside, our Shannon Ferris, who's hosting or chairing our next session, when she was in my lab, really did a nice study with RNA-seq, and again, please play around with this. It's a really good way. If you have a gene of interest, you can see where the actual expression levels in more detail than maybe what the Allen Brain Atlas in situ hybridizations will give you. So the work I'm going to talk about is the one published in Hippocampus. This was led up by Dan Radzicki in my lab and Sarah Chong. And we thought we would just actually try to see how these stains matched up. We used RGS14 and PCP4 antibodies against these proteins because they really do seem like they, well, first of all, they're commercially available and they work really well in a number of species. And so we thought this would be really useful to see exactly how they overlap. And so throughout this part of the talk, you'll see me referring to this yellow portion. Actually, it's not on this slide, but we call this the dual labeled zone or double labeled zone, just for lack of a better, definition. And so if we look on the CA1 side, this is CA1, this is this double labeled zone, and these green neurons are PCP4. And if they're double stained with RGS14, then that's the double labeled zone. We see that the extension of these kind of straggler cells labeled by PCP4 that are RGS14 negative really do stretch into CA1, and whether those are CA2 neurons, I guess we can talk about that another day, but they seem to be located in CA1. And the number of those cells really increases, or rather the length increases as you go more ventral in the hippocampus. And the flip side here, RGS14 is the one that kind of trickles more into CA3 a little bit, but doesn't really differ from that of PCP4 and doesn't seem to be, doesn't seem to go into this, that doesn't seem to vary by dorsal ventral axis. Again, here it's where we double stained just a little more here, that they don't always overlap exactly. And again, actually, this is just the same figure and some more detail here. And again, please go to the paper. So again, this dorsal ventral axis really is driven by this increase in length in the PCP4 staining along the dorsal ventral axis. Okay, so this double labeled zone overlaps a lot with some of our other stains and also our Amigo2 GFP, but they all differ just a little bit. And, so when you're choosing, maybe you don't have the option to double stain, but maybe you want to use STEP. STEP is a good one. It's slightly bigger than the double labeled zone, but it's also overlaps with a lot of these stains. And again, in this case, step trickles more into the CA3 side. So again, it might depend on what your questions are. So if you're wanting to define the CA1, CA2 border, maybe you're not wanting to go with, maybe step is a good one. And alternatively, and for the CA3 side, then PCP4 might be the better option. VGLUT2, these are those SUM axons. Also strong overlap with this double label zone. And again, what this is showing here is that it does fall a little short on the CA1 side of the double labeled zone. So maybe this is, these are where the VGLUT2 staining length falls short of the double labeled zone. This doesn't seem to vary across the dorsal ventral axis. And now some of us make use of the Amigo2 GFP mouse, and also Amigo2 to drive the Cre recombinase. How does that line up? And consistent with some previous work, it's a little bigger in both sides. That's what's showing here. This one does seem also to creep into the CA1 border more in the ventral also. So now the thing is with some of these lines, it doesn't mean that they're expressing these genes as adults. Maybe they're developmentally regulated, but it does really point to some heterogeneity among these cells within CA2, and I think we might be hearing a little bit about that. But I did want to go back to this idea of, oh, sorry, this is the WFA. This deserves a special mention. This is to label the perineuronal nets. Now, this really is not a good marker for CA2. It nicely labels CA2, and it's depending on what your microscope settings, you can get really a nice CA2 stain, but it really is denser in what might be considered the classically defined CA2. That is those just beyond the CA, the mossy fiber terminals. I'm going to talk a little bit here also about this strongly differs between different species and even among different species of rodents. So even though it's a really interesting stain and that it seems to be activity regulated, we had a paper, it's actually our Rett syndrome paper, where Georgia and Kelly in my lab had silenced or increased an increased activity in CA2, and actually it seemed to be inversely related to activity, to neuronal activity. So when you had GI dreads, we got an increase in the perineuronal nets, or the GQ dreads led to a decrease. So these are activity regulated, and so maybe this suggests some sort of that these cells might be less active, but I don't think we can say this at this point, but it really, the strong difference between species is what I would argue is maybe not the best marker. Okay, so mossy fibers are distinct in CA2. They tend to be smaller than the boutons. This was consistent with what Kohara had reported. But what is super interesting, I think, is that these axons, it's just worth repeating because I think it's an interesting finding, is that the dentate gyrus axons come around the curve of CA3. So this was an injection, the red into the dentate gyrus comes around, and we can see, of course, them in CA3. But then if we go a little bit more ventrally in another plane of section, we see that that red stain, that red protein, I think it was TD tomato, is really only in CA2. And this really implies that these axons are coming around the curve and then shooting backwards caudally or ventrally, if you will, within CA2. And so that's, I think, a super interesting feature that we were able to replicate from Kohara's lab. I mentioned the mossy fibers, the thorny excrescences that are associated with the mossy fibers. As it turns out, they're not a defining feature of CA2 pyramidal cells. So if we look here, this is CA3. In this, you see a lot of these mossy fiber, the thorny excrescences on the CA3 cells. But well within what I would call CA2, we occasionally do see these thorny excrescences. They're not real common. For the most part, these cells are lacking the thorny excrescences, but they're there. And I do remember Rebecca mentioning this to me I think it was the Hippocampus meeting probably 4 years ago or so, that there were they saw thorny excrescences in CA2, and I'm like, oh, that's ridiculous. But we thought we should, well, we saw that ourselves. It was not something we set out to see. Okay, so that's mice. We getting just a little bit into the function, or rather what we might be able to infer from the anatomy. We were interested in, or I think my lab thinks I'm a little nuts on this, was wondering if foxes have a CA2. And the reason for this is that there's evidence for CA2, a role for CA2 in aggressive behavior. And this was starting with work from Scott Young's lab in the vasopressin receptor 1B. Knockout mice had reduced social aggression. And of course, some of the nice work by Felix Leroy when he was in Steve Siegelbaum's lab had shown that CA2 activity modulates aggression. And I simply wanted to know, can we learn something about CA2's role in aggression by looking at foxes? Well, why foxes? Well, there's this really cool, I would say, ongoing study that it was started in Russia since 1959. So this is the Russian fox farm experiment, where the investigators started breeding these animals to be tame, selected for tameness. And if you ever want to get online and just see some of the videos of these animals, there's a group outside of San Diego that they're adorable. Now, they're not affectionate in the way that dogs are, and you can see some of the videos, but they'll approach humans, but then, you know, they're not all that interested, but they're certainly tame. They've, as a later part of this experiment, they started breeding some animals to be aggressive. Now, why do I think that CA2 might have anything to do with this? Well, in a nice study, fairly recently, was shown that the hippocampi were among the areas of the brain that were enlarged in both the tame and the aggressive animals. So we want to know if there's something special there. And spoiler, I haven't gotten to that yet. We first thought we needed to define CA2, and this wasn't at all obvious where CA2 was in the dog. So, or in other species like dogs, like cats, like ferrets. In fact, a number of investigators said that there's a CA2 in the dorsal, but not the ventral. It's just been very confusing. And these studies are mostly based on missile stains. But they did have the mossy fibers stained with the TIM method. We did not have that luxury, but I thought we'd start with some staining that was similar to what the other studies had shown. with the Nissl. And I think the CA1 cells could be identified with some confidence, but really you'd be hard pressed to say what the difference is between this neuron, these neurons, or is this all CA2? Is this CA2? This looks like CA3, I can say that with some confidence. But I think it's with Nissl stain, it's really not possible. And this would be, I think, also evident with the fluorescence Nissl, and that has allowed us, and I think that if the reviewers out in the audience, I thank them for the suggestion. I didn't know you could do a fluorescent Nissl, but if we were to guess, we'd say the mossy fibers end right about there at that little niche, and that might be CA2. What do you think? Yes, that's exactly the case. So I think in foxes, there's a lot of similarities, homology, whatever you want to call it, with the mouse, in that a good portion of the PCP4 positive cells do receive something that could be the mossy fibers, and some of them don't. Now, these might be these other straggler cells that I was mentioning that we also saw in the mouse, because we also saw a lot of them in the ventral, and I'll show you that another slide. And I'll try to wrap up here in a minute, but we also used, like I said, we couldn't really label the mossy fibers with the TIM stain, but we did use calbindin here, and we could see pretty well the end of the mossy fibers. Unfortunately, the CA1 cells, which also are positive in a number of other species, kind of get in the way of really being able to tell the end of the mossy fibers. So one of the things I had noticed when I was looking at the WFA stain was that we kind of have a negative relief, a negative stain for the mossy fibers. And so in the next slide, I'll show you, you'd be using that. And again, as I had mentioned, this WFA stain really is not selective for CA2 in this species, apparently, in that much of CA3 is also labeled with the WFA. So here is the WFA stain with the PCP4. And again, you can see these are outlined with the WFA and all of these areas here. But notably, we do have a PCP4 stain, and this is really deeply ventral, as far back as I could cut the sections. And I should add that I pulled the hippocampus out and cut this exact as close to perpendicular to the long axis as I could. So these are what we would call the coin cut of the hippocampus. And you see that even as far down or back as you go, you still see a PCP4 stained population. And just
Serena, we're gonna I don't want to cut you short, but
okay, I'll wrap up.
We're at your time limit.
Okay, I started late. I got a couple more minutes. Okay, only to say here that when we could double stain with PCP4 and calbindin, because this antibody just recently became available, we see some really clearly calbindin negative cells with the that are probably the interneurons, the calbindin positive interneurons. And we see some really clear PCP4, most cells are PCP4 positive and calbindin negative. But just to throw this out there, we did see some that lightly stained, double stained for both. So this does tell us there's maybe some heterogeneity. And just to shout out, Preston in my lab has been staining vole and hamster. I should say PCP4 and RGS14 both stain those very nicely. And to conclude then, a lot of these stains overlap. They overlap a lot, but not exactly. And these two stains are really good in a number of different species. The WFA, again, I would argue is maybe not such a good marker for CA2. Also just on an individual cell level, I don't think that the thorny excrescences are exclusively in CA3. All right, postdocs wanted, and I'd like to thank my lab.